All right. Hi, guys. I hope that you are all doing well. Couple of general announcements before I get started. One, mostly because I don't know how to say the word mishmat. The word I still can't say. We're just going to talk about one topic today and not put two together. So it's just going to be no, mishmat. Yeah, that one. Yeah, that word I can't say. Anyway, we're just going to talk about cancer immunology today. Um, you got a doodle poll from me, and I asked you to reply before today's class in terms of times for a review session for the final. The most popular time on the doodle poll was Saturday at 10. So that is when we'll have the review session. I will send you guys a link to a Zoom for that, and make sure you have your questions. And remember that the final is on Monday at 12:30. Also remember that you have some homework due on this coming Friday, so make sure that you're ready for that. Today we're going to continue talking about cancer immunology, leaving off from where we were last time. And so what I want to just sort of briefly remind you is that there was this controversy in the field for a while about whether or not the immune system did anything about cancer, based on some experiments, like the experiment shown on the right that I told you about last time, as well as based on observations of immunodeficient mice and immunodeficient people who got all sorts of tumors. People realized that, in fact, the immune system does do something about tumors. I'm going to harken back to one thing that shows up on this slide a couple of times today, so I just want to point it out now, because it does come into one of those kind of places where people were arguing for a while, and one of those points of contention, which is that if we look at our original mouse that was treated with some chemical carcinogen to induce a tumor, one of the arguments for cancer immunology being a thing was that we could do things like take the tumor out of this mouse, or cure this mouse, put a tumor back in, and the mouse would reject the tumor. Or we could transplant CD8 T cells, and those could allow for rejection of the tumor. And so these types of experiments let people see that the immune system can reject a tumor. But if we had this mouse with a tumor who was just sitting around hanging out in the mouse colony, it would keep its tumor and probably die from that tumor. So there's this idea of the immune system can deal with these tumors, but people still end up dying from them, and mice still end up dying from them. So there's also this idea that the immune system doesn't always do something, and the tumor generally wins. And so this was one of those places where there was some uncertainty. I showed you some of the data that kind of convinced immunologists that the immune system could do some things about cancer. And if you remember, one of the big questions that I mentioned to you last time had to do with the antigen issue. The idea that, tumor cells are self cells. So how the heck are we getting a anti-self response when just, 2 lectures ago we were talking about autoimmunity and not wanting to get an anti-self response? And we talked about the 3 different types of tumor antigens that we might see, either tumor-specific antigens that come up when we have mutations, or other things that are overexpressed, or inappropriate expression of normal embryonic genes. And so all of this information that I've kind of started to tell you about last time actually led to this idea of cancer immunology as a field and the beginning of our understanding of tumor immunology. And one of the things we have realized in the time, in sort of over more recent years, is that when I told you about the 6 hallmarks of cancer last time, sort of the 6 things that a cell must be able to do to form a tumor, that there actually is something else besides that. In fact, in reality, in the field, there are more than 6 that people talk about. We're just going to have a 7th one. And you can take Dr. Dunaway's class and learn about more kinds too. And so your textbook shows these 6 hallmarks of cancer as things that all cancer cells must accomplish. But immunologists have also realized that tumors actually have to be able to evade the immune response or otherwise outrun the immune response. And so actually being able to avoid these responses that I've shown you can happen is a key thing that tumors must be able to do. In understanding this issue where all tumors need to evade the immune system, immunologists have come up with a theory about how the immune system interacts with tumors. The theory has 3 parts, and so I'm going to tell you the overall name of the theory as well as start going through the parts on the next slide. I think in the past, sometimes students have gotten confused about what was like the big picture name and what were the 3 individual steps. So make sure that you're noting those differences. And so we're going to be talking about this theory, which is known as the cancer immunoediting theory. It's a 3-step process that is really important for how tumor cells interact with the immune system. And the first step is, you can think of almost like as a first phase, what's happening very early in the progression of that tumor. And this is known as the elimination phase. The idea in the elimination phase, and I've got figures from 2 different textbooks here to try to give you 2 different ways to visualize this, is that we'll have some cells that have acquired some mutations that have perhaps become transformed, and that they will be attacked by cells of the immune system, particularly T cells like CD8s, CD4s, and gamma deltas, but also by NK cells. It can be by a number of different cell types, and they will be killed and, in fact, eliminated. That's how we get elimination in the name of the phase. And you can see that happening over here as well. We normally have some of this healthy tissue that's orange. So we have some cells that may pick up a mutation and be transformed cells that are sort of this nice pink cells. And we may then eliminate those pink cells with our cytotoxic cells of the immune system, getting us back to nice, normal, healthy tissue. And so you can sort of imagine, well, okay, this is almost like, shall we call it like a microtumor or sort of the beginning of a cancer in a patient, and the immune system wipes it up. And you can imagine perhaps this is happening really frequently in people, and the immune system is kind of keeping us cancer-free all the time. As little tumors start to develop, the immune system gets rid of them very quickly. However, as time goes on, things begin to change. And I would say I kind of think about where the place that we're talking about in terms of a tumor is like way back here, way at the beginning, before we're really getting those population doublings, before we're really even getting that cell to kind of take off. So we're way before that growth of a tumor that I showed you about before. We're way in this early time period before our patients are potentially out to tumors. And that cell that got eliminated maybe is like this cell early in the process that had picked up a couple mutations. What you might notice is that it maybe still had a little bit going wrong with a precursor cell. There might be some other cells around that still had the 1 mutation, not just those 2 mutations. So things are not like all perfect here. But we've gotten rid of the cells that have actually gotten to the point of having that tumor antigen. However, because we aren't, with some perfect cell, the cells can continue to pick up more mutations. And that cell can continue to mutate, and that cell may mutate such that it starts to avoid the immune response. And Now we're going to see what's known as equilibrium, where we're not going to get a huge increase in the number of tumor cells, but we're also not going to completely eliminate them. The immune cells are going to be able to get rid of some tumor cells, but not all of them, and we're going to kind of get to this equilibrium phase. So here you can see originally the immune system can eliminate all the tumor cells, but we're going to continue to get some variation as those cells pick up mutations, and eventually we may get some variants of those tumor cells that are more resistant to being killed. And so we're going to get this sort of equilibrium where we have some tumor cells and some immune cell killing. And you can see, again, this process of equilibrium here where we're getting kind of this increased number of these tumor cells, but they're not really sort of taking over. We're at this equilibrium point. Eventually, those tumor cells are going to keep picking up more mutations, and they are going to be able to escape the immune response. And so we're only going to get to the point where we actually see that cancer progressing, and we get to see kind of a larger tumor with a larger number of cells when those cells have picked up mutations or figured out some mechanisms where they can actually avoid the immune response. So there's this third phase of escape. And so what this sort of means is that usually for these, tumors that are getting to the point of being visible on an X-ray or palpable, they have first had to spend quite a bit of time, picking up mutations and also trying to avoid the immune response and escape. And so before we can even get to, 1 doubling or 10 doublings or 30 doublings that get us to a very large tumor, we actually have to get the ability to evade immune responses. And that, as I mentioned, is listed now as one of the hallmarks of cancer. So that's in and of itself kind of, interesting. But we as immunology students need to think a little bit about how this evasion happens. What specifically are these cells doing that allow them to evade immune responses? And the reason why this is important for us to think about is that if we can understand how these cells are evading immune responses, we might be able to turn the immune responses back on or somehow unevade the responses. And so if we can understand what's going on with this evasion, A, we're going to learn some kind of interesting immunology, but B, we might come up with some places where we can intervene by trying to stop this evasion and let the immune system do what it could do at one point when it used to be able to eliminate the cells. We can now let elimination happen again. There are a bunch of different ways that tumors are able to evade immune responses. And so I'm going to list some of them here. Different tumors will have different evasion strategies, really based upon which genetic changes they acquire. One of the things that we will see in many tumor cells, like this tumor cell you can see here, is that tumor cells will usually start expressing less MHC, particularly class 1. And so you can imagine that if we have a tumor cell that has lots of class 1 on its surface, it's going to be presenting a lot of the tumor antigen, and that cell is going to get killed pretty quickly. And so we might have some cell that is going to express less MHC. Perhaps there is a mutation in the promoter of MHC, so we make less of the MHC protein or something involved in the antigen presentation process. Lots of options. And so those cells are going to have less and less MHC, and so they're going to be more difficult for T cells to recognize. Here you can see a tumor, and the brown is MHC staining. Remember, when we talked earlier in the semester, we talked about the fact that all nucleated cells have MHC. And you can see that there's an awful lot of this tumor that has no MHC, no brown staining, because that tumor has actually lost MHC class 1 presentation. So that's one way that some tumors will be able to evade. When I showed you this, at least one of you made a face. Because when we've talked about MHC class 1 presentation before, and cells not having MHC class 1 presentation, we also came up with another solution. So if you look at, if it was just about loss of MHC class 1 presentation, what might happen immunologically? Or AKA, why did you make a face? Yeah, Grace.
The NK cells would kill them?
Yeah, one of the biggest points of NK cells that we talked about is that NK cells recognize cells that are missing MHC class 1. So if we have these cells who are missing MHC class 1, oh, cool. They can't be killed by CTLs anymore, but they can be killed by NK cells, so that's easy. So one of the other big things that tumor cells will do is they will actually stop making NK activating ligands. So oftentimes, remember when cells are stressed, they will make certain proteins, like the MYC proteins, that can bind to NK receptors and activate those NK cells. Tumor cells can also acquire mutations that will allow them to stop making MYC or instead make a version that's cleaved and that can totally soak up the NK receptors, so that now these cells are not able to be killed by NK cells. And what you should imagine here is that, there's this tumor cell and its progeny are each picking up some different mutations. It's not like the cell is maybe directing the mutation to happen in the MYC gene, but if a progeny happens to stop making MYC, that progeny cell is going to have such a selective advantage because it's not getting killed that we're going to see those tumors really have the ability to outgrow compared to other cell types. We can also see things like, before we talked about, well, let's get rid of MHC and stop presenting on MHC. Sometimes this tumor cell actually stops making the antigen. And so instead of changing MHC levels, we can have tumors that will stop producing their antigen, that tumor antigen, so that, again, there is nothing for the T cell to recognize. And we talked a little bit about how dependent this is on the antigen last time, because these are self cells. We can also see situations where, and this is actually pretty common, tumor cells tend to turn on immunosuppressive proteins. The most famous one being PD-L1 that happens to interact with PD-1 and be a negative co-stimulatory molecule. And so tumor cells can basically turn off any immune cells that come into their area. This can also happen via immunosuppressive cytokines like TGF-beta. And so the tumors can actually have this immunosuppressive environment, sort of make any T cells that come near them get polarized towards Tregs or otherwise get turned off, and stop those cells from further activation. You can see they're very wily, these tumor cells, with all of their ways of evading. And some of them, in addition to producing all of these different immunosuppressive proteins that you see here, some of them can even start to build a physical barrier and make themselves their own immune privilege site. And so actually induce basically a physical barrier to keep the immune system out. And so pretty much all of the major things we've seen that tied into immune tolerance, and if you think about many of the mechanisms of peripheral tolerance that we talked about earlier this semester, they tend to be things that tumor cells have evolved to do in order to allow the body to be tolerant of them. One other piece that comes in here is related to the timing that you see on the right of this slide in terms of tumors tending to be seen in individuals who are older. And if you imagine this requiring many, many doublings of a cell, and in fact, the cell going through elimination and equilibrium and escape, and then all the doublings, you can imagine that takes a really long time. And even if we just have that one tumor antigen, that is being presented normally over 20 plus years, we can also realize that this is a prime situation for T cell exhaustion to happen. And so sometimes it doesn't even have to be as completely active on the tumor's part, although the tumors often are doing active immune suppression. We can also see that T cells are, as they are seeing persistent antigen for years and years and years and years, may move towards this exhausted phenotype. Remember, the exhausted phenotype does have high levels of PD-1 on the surface, as well as lower abilities to do a lot of different functions. And this is the same exhaustion slide we've seen previously on this semester. So at this point, kind of, we can say, all right, we know the immune system does do something about particularly really early, microtumors, and that all tumors had to figure out ways to evade the immune system. But then we can also think about, well, can we use our knowledge of immunology to combat cancers? And so it's sort of the other side of this same question. And in one way, the answer is like, yeah, duh, really easy. We talked about the fact that some cancers tend to be caused by certain microbes, by certain pathogens. This is a case particularly thinking about HPV, human papillomavirus, and things like cervical cancers. And if the issue is a microbe is causing the cancers, then it's really obvious how we can use the immune system to deal with that. We can just vaccinate people so they don't get the microbe. And that solves everything. And so this is showing a little bit of information about the HPV vaccine. We're going to talk more about vaccines in general on Friday. But this is able to protect against 70% of cervical cancers, and in a more modified version, 90% of genital warts. Here we can actually see antibody titers and months, and you can see people who get the vaccine make a good antibody titer to this virus, and thus do not get infected with this virus and are thus protected from cervical cancer. So in that case, we're like, yeah, of course we can use the immune system to combat cancer. But we can also think about kind of other cancers, particularly situations where we're not thinking about viral infection. When we think about some of these therapies, I want you to remember that we're going to, in some cases, be contrasting them against things like radiation and chemo that might involve bone marrow transplants. And so we already talked about radiation and chemo and some details of how they might work when we talked about bone marrow transplantation. And so remember that these therapies can deplete rapidly dividing cells, but they also basically remove the entire immune system. See, the person's empty. And so we have to use a bone marrow transplant to reconstitute the immune system. You can see your textbook tells you many kinds of cancers that are treated by bone marrow transplantation. So the idea is that this is awesome because it kills the rapidly dividing cells, but it sucks because it kills the bad rapidly dividing cells of the tumor, along with the good rapidly dividing cells of the immune system, as well as a number of other anatomic locations. And so what we would really like to be able to do is narrow down this therapy and try to target just the tumor cells and not all the cells altogether. If you think about what we talked about with autoimmunity treatments, this might sound familiar. And how did we start to think about making this work with autoimmunity treatments? We just talked about it like the other day. Remember, we were going from a narrow cyclosporin A, or a broad cyclosporin A, to more narrow therapies. Yeah, Emma. We use antibodies. We took advantage of the specificity of the adaptive immune system to try to more narrowly target cells of interest. And what can be useful in this situation is that here we've identified some antigens. We actually have talked about some tumor antigens. And so we could even say we need to eliminate those cells with those particular tumor antigens. And so we can do kind of the same things conceptually of taking advantage of the specificity of the adaptive immune system. And so one of the big ideas that people first came up with is shown, has versions that are shown here. And the idea is basically to take an antibody and link either a toxin that could otherwise be like a chemotherapy toxin or a radioactive molecule to the Fc portion of that antibody. And you can have that antibody binding to the tumor antigen. And so now we're delivering our toxin or our radioactive molecule directly to the tumor cells and not broadly to cells throughout the body. And so you can see this, these antibody-directed therapies here. You can also see that sometimes we even will just use an FAB and attach our toxin to the FAB to have a smaller molecule that can specifically kill the cell. And so the pharmaceutical industry really likes these types of molecules. They're called ADCs. And they are things that people are doing a lot of to actually try to specifically target those molecules that we were using before very broadly and kind of hitting the whole body with, to just try to hit tumor cells instead. In starting to think about this, well, immunologists also realized something else. So these are amazing, and lots of pharma companies are working on these, a ton. But the other thing that people realized is you actually probably don't necessarily have to put something on the Fc portion of the antibody. If you have antibodies that will bind to your tumor antigen, and so again, this and every other thing I tell you about now does require you to know some things about a potential tumor antigen, maybe a protein that is often overexpressed in a group of tumors. If you just can get your patient to have antibodies against the tumor, your immune system can do the rest with all of the things that we've talked about all semester in terms of antibody effector functions. And so the original idea was we're going to put toxins on and deliver those toxins uniquely to individual tumor cells. But since that time, people have realized, actually, if we just deliver the antibody by itself, with nothing attached to the Fc portion, we can have things like NK cells come in and kill that tumor. Or we can have something like macrophages interacting with that tumor, or a number of other effector functions, all the effector functions we've been seeing all semester. And so this is a figure and a table from your textbook kind of showing a number of these different antibodies that people are using. Here you're just seeing a general example of using this antibody. We don't have any special payload on the Fc portion. We're just letting the NK cells do their job. And here on the right, you can see a bunch of different antibodies like this that are used in cancer therapies now. Yeah, Josh.
This is what the talk was about, right? About a certain antibody.
This is what the talk was about. So yes, I was going to mention that in just a second, but excellent job reading my mind. And so you can see that there are either signaling pathways that are known to be highly expressed or overexpressed, or other particular antigens that are unique to a particular tumor type. And the idea is we're just using the antibody effector functions that we saw earlier this semester to allow the immune system to do its job. As Josh mentioned, the talk that we had earlier this semester from the RISE group, from the group run by RISE, actually involved an antibody like this. Their antibody was pretty cool because it's actually called a bispecific. So it has 2 different FABs and combined to 2 different antigens. That allows this to be even more specific to a tumor cell, because you might have situations where you might have some of these proteins on healthy cells, but in fact you're trying to get, this antibody will only bind if both proteins are there, so it makes it more specific. And they showed in the talk that this both involves the cell turning, or basically reducing the amount of antigen it makes, and this antigen leads to some signaling that's problematic for the cells, so that's good. It also induces the tumor cell to be killed by NK cells, and it induces a pretty cool type of macrophage interaction with these cells called trogocytosis that I don't have time to get into, but it's so cool here. And so it's exactly the same kind of idea of just taking advantage of our knowledge of the immune system and using antibodies to give us some specificity in attacking these cells. There's one other antibody that's used really, really frequently for a lot of cancer therapies. And that is an antibody that specifically is going to bind to one of our negative co-stimulatory molecules. Here, your textbook shows this being an antibody to CTLA-4. The image that I have here on the bottom, make easier to see, shows that antibody interacting with PD-1. Remember, PD-1 is that protein that's involved in T-cell exhaustion. And so either of these things, either stopping CTLA-4 signaling or stopping PD-1 signaling, will stop T-cell exhaustion. And so our T-cell that was previously able to fight the tumor cell and then got exhausted can now potentially get turned back on. We can reverse the T-cell exhaustion, take those T-cells that did a good job eliminating potentially a couple phases ago, and turn them back on and let them start elimination of the tumor again. So all of those things are some pretty cool things that people have thought about. There also have been some ideas that a number of immunologists have had about trying to have some tumor vaccines. And so, we can do this in animals, like in mice, pretty easily. And I kind of showed you this in some of the previous experiments. So we might have, we can have a situation where we might, give our animal a vaccine with a particular tumor antigen. And then we might later challenge our mouse with the tumor cells, and hooray, we get no tumor. And we can also, in fact, just transfer CTLs and get this same protection. So this is great in our animal models. But there's a little bit of a reason why this is a little tricky with people. So in this mouse, I can give this mouse this tumor vaccine to prevent the tumor and happily protect that mouse. I have saved that mouse from this cancer. Why would it be really hard for me to do the same thing to you? Yeah, Joel. T cells are pretty good, so we probably doesn't have something to do with the difference in relative size. But let me give you guys a hint. If I were going to make this actually work in you, one of the reagents or pieces of lab equipment I would require is a time machine. So look at this. We gave the mouse this tumor vaccine before we gave it the tumor that I knew I was going to give it. Yeah, Josh.
Because we want to, if we remember what the adaptive system does, it's like you see it, it's very important that you see it the first time, right? So if you can't give us antigen because you can't give us a vaccine for antigen we've already seen, right?
Well,
I feel like it's along the
so the big question is, I don't know what tumor you're going to have in 20 years, so I can't give you a vaccine with that antigen now. And by the time you actually present with to a doctor and have that tumor, you've already gone through those elimination and equilibrium and escape phases. Your immune system has already, you've already had this escape. If I could know in advance the tumor that you're going to get and what antigen that tumor would have, then sure, I could vaccinate you against it and we'd be good. But I can't know that, right? That there's sort of this problem of I can't know what the tumor is. But yeah, we can show that we can do these kind of vaccines. The problem is that if I wait till you have the tumor and I take the tumor out and I try to make a vaccine with it and then I put it back into you, what's going to happen? So you have a terrible tumor. I take it out. I, irradiate it or something, and I inject it back into you. Yeah. You still have the tumor, and the T cells are already exhausted. The whatever evasion mechanisms have already happened. The immune system's already kind of unable to fight that tumor. So if I want to actually be able to get you immune responses that fight against that tumor, I've got to be able to sort of help make those immune responses happen outside of your body, because there's already weird immunosuppressiveness happening in your body. And so that leads to this as one thing that we sometimes will do. Sometimes we'll take a patient and we'll actually take out their tumor or parts of the tumor and find the antigens, and then also take out some T cells from that patient, some PBMCs, and culture the tumor and T cells in a dish and get good active T cells in a dish that aren't dealing with all of that immunosuppression and weirdness of that tumor environment. And then we might take those T cells and re-inject them into the patient. And so now we've tried to sort of get working T cells in vitro in a dish to allow our patients to have an immune response. And so we can use sort of some other parts of our knowledge and the fact that we know the problem is this immunosuppressive environment that's happened in the patient and try to make this work well. And here you can see sort of some data on this. And you can see that when we transfer in the T cells, our patient's white blood cell count improves a lot. Our patient actually does quite well with this type of thing. But you can also imagine, like, this means that for every single cancer patient, we have to take some of their tumor out, get some T cells, grow them up in culture, get them to work, put them back in. That's pretty expensive. That's pretty, hard to do. You can imagine it's not having, both part of your tumor taken out and donating T cells. Oh, that can be a bit difficult for many of our patients, but it has been really, really useful. We've also seen situations where we can actually take out innate immune cells like monocytes from our patients and culture the monocytes with cytokine and antigen. So they're really good at presenting those tumor antigens and put those back into the patient with the idea of basically getting sort of antigen presenting cells that are awesome presenters to try to help out a lot of those patients' cells. And so this is something else that a lot of people have thought about. And one of the other big therapies that people have spent a lot of time thinking about is something called CAR T cell therapy, or chimeric antigen receptor therapy. So I'm going to tell you kind of some of the steps of chimeric antigen receptor therapy. We can talk about some of these first particular patients. And so we have a patient here who is our cancer patient. And the first thing that we're going to do from this patient is we are going to take out some immune cells. So we're going to take immune cells out in the blood. Particularly, we're getting T cells. So you can see we've taken out the patient's T cells and we've grown them in a dish. You can also see on this slide this picture of a little girl. Her name is Emma Whitehead. She was the first patient of CAR T cell therapy. And I'll say some other stuff about her later. But this figure from this textbook actually has this little kid picture of her here. And so once those T cells have been taken out of the patient, these aren't necessarily T cells that are acting against the tumor. These are just some T cells from the patient. We modify those T cells in the lab. And the specific modification that we do in the lab is that we insert a new gene into those cells. And we're going to use a virus to insert that gene into those cells. You can see that happening over here on the right as well. So basically, we're using this gene, we're inserting the gene into those cells to allow integration of this new gene into those cells. All in the dish, not in the little girl. And the specific gene that we are inserting encodes a special receptor known as the chimeric antigen receptor. The chimeric antigen receptor has a few different parts. One part of it is actually the variable region for an antibody. And that antibody is known to bind to the tumor antigen. In the case of Emma Whitehead, the tumor antigen was CD20, which was on all activated B cells. And so they used the variable region of an anti-CD20 antibody. So if you looked back at that antibody slide I showed you before, the CD20 antibodies are used for a number of different patients. But here they take the variable region of that CD20 antibody and they put it onto some domains from some other proteins. In particular, you can see we've got some domains from CD8, and most important for the stuff that we've talked about, we've got some domains from CD3 zeta. Specifically, those domains from CD3 zeta are the ITAMs. And so basically what we've done is we've put in a receptor piece to this that binds our antigen of interest, which is the tumor antigen. And then we've put in the rest of the protein that actually allows for all the parts of T cell receptor signaling to happen downstream. So this is a figure from your textbook. You can actually see there usually is a CD28 domain. We've got some of those zeta ITAM domains. We basically have domains to allow every single part of T cell receptor signaling, including all the signal 1 and signal 2 stuff that we learned about this semester, to happen from this one receptor. And so now we've taken T cells from our patient, and they might be like a random influenza-specific T cell. But we've inserted this new receptor into them. So yeah, they still have their influenza T cell receptor. But they also have this new kind of Franken receptor that binds to tumor antigens and can do all of signal 1 and signal 2 signaling. And so if that T cell ever sees the tumor antigen, it can automatically be turned on and automatically act against the tumor cell. And so it's because we're putting together a lot of different pieces of different receptors, we call it a chimeric receptor to bind to our tumor antigen. So that's where chimeric antigen receptor, or CAR, comes from. Once these are made in the dish and sort of grown up to larger numbers in the dish, we actually will then put them back into the patient, with the idea that now the patient has activated T cells that we have activated in the lab that bind specifically to their antigen of interest that can start to deplete this tumor. And so I show you up here on the, and so you, well, let's see. Here you can see the same thing. You know, our T cell still has its T cell receptor that bound to whatever, maybe influenza, but it also has these chimeric receptors that we've put in. And so if those receptors ever come across the tumor cell, now our T cell is going to kill those cells. I mentioned this little girl, Emma Whitehead, who was the first person treated with this. She got some headlines in the news earlier this semester when she started college. This is from her blog, and this is from the Philadelphia Inquirer, that she's actually going to the same college where they did her treatment at originally, now 10 years later, because this has worked really well. It was touch and go for a while. When they did the first infusion, she had a massive burst of IL-6, and they had to use a whole bunch of IL-6 depleting antibody to bring her IL-6 levels back down. That's something that now they kind of know to look for and do earlier on. But this seems to be working, extremely well, and to be really useful for the patients. One thing to realize is that in this therapy, Emma Whitehead got these CAR T cells that had a chimeric antigen receptor that bound to CD20, because she had a B cell tumor, and CD20 killed and CD20 is on the particular B cell tumor. What's the problem still with this therapy? Yeah, Andrew.
You need to know what the tumor is specifically, and then make T cells proliferate them in a way where they target that tumor.
So we do specifically want to know some things about the tumor and be able to target that specific antigen. And here the scientists had done that, and they knew that CD20 was on the tumor. But there is another little bit of a problem. Yeah, Grace.
CD20 is also on all the other B cells?
Yeah, all the other mature B cells also have CD20. And so you can see this is an amazing therapy here, but this therapy also killed all of her other mature B cells that she had at that time. What's certainly better than killing every rapidly dividing cell in her entire body, but it still, has some work to do and has some improvements. In 2 different conversations I've had with people from the pharma industry within the past month or so, they've both told me that there are lots of questions about CAR T cells right now, and sort of how well they can be infused into patients, and sort of how much people might use them in the future. But I will just tell you a little bit more. CAR T cell therapy was the first ever gene therapy approved in the United States. And so you can see that approval from 2017 listed here. So you can see the FDA approved Kymriah for certain pediatric and young adult patients with acute lymphoblastic leukemia. This is exactly what she had. And you can also see that this was listed as the most expensive perhaps therapy that had been described, being listed that just making the cells alone for one patient was $475,000. And then, of course, things like the infusions and all of that and other parts of the therapy will put this therapy well over $1 million. So something to be aware of. One of the things that people are also thinking about are ways that they can improve CAR T-cell therapy. And I'm going to mention 2 improvements on this slide and the next slide. But one of the things that I think is really interesting and ties again into what we've been thinking about a lot this semester is that a lot of individuals are thinking about whether they can make CAR T cells with multiple receptors, just like you saw with that antibody from the talk earlier. Potentially, a tumor might have more than one antigen, might have the green square and the purple circle, while normal cells might only have one or the other. And so perhaps if we make a CAR T cell that has to bind 2 antigens, it's going to become more specific to just the tumor and less and kill fewer of the normal cells. So maybe if we found a second antigen, like Andrew mentioned, on those B cells, and we could target CD20 positive cells that also had this other antigen, we could kill just the tumor cells and leave the other mature B cells alone. And so ideas about having kind of these multiple receptor circuits are a big place where a lot of immunologists have been going. And at least one review that I've read about them called them trucks because they were trying to make a play on car, and I was not a fan. One other thing that I've been learning a lot more about is that a lot of people are actually realizing, why are we taking the cell that we're modifying and using a T cell? Why not put the receptor into something else? And so there are now things like CAR NK cells and CAR macrophages that have different properties of attacking those tumor cells and may actually work a little bit better. And so whether we need to go with CAR T, originally people were like, we're going to go with CAR T because like we want to kill things, and cytotoxic T cells are what kill things. But now we've realized that a lot of the effector functions of some of our immune cells might be important here. So this is actually a podcast episode that we did talking about CAR NK and CAR MAC cells versus CAR T cells. And this is another one we did just about CAR T cells more specifically. And so thinking about, does it make the most sense to take advantage of a T cell effector function, or should we be using a macrophage effector function? Should we be using an NK cell effector function? Which cell should we be trying to move to the tumor cell? Which cell might be able to best overcome the immunosuppression of the tumor environment? And which cells are going to give us the best effects with the least side effects have been a big area that people are really thinking about looking at. So I want to tell you about one other thing. I feel like if this were Dr. Dunaway telling you about cancer immunology, he might end here. Not likely. But there's one other thing that I think is just so cool that also ties into this that I like to mention. And so the other thing that I like to mention here is something called oncolytic virotherapy. I think is so awesome. And again, remember that our problem here has been in having therapies that are very broad and sort of wanting to narrow them down. We know that viruses are very picky or very selective in terms of which types of cells they infect and reproduce in. For example, you likely know that poliovirus can replicate in neural tissue. Or you might know that certain pox viruses that give you a rash replicate in cells of the skin. And so the idea with oncolytic virotherapy is to actually come up with a virus that can infect tumor cells and only replicate in those tumor cells. And the original idea with oncolytic virotherapy is that you would have a virus, it would infect the tumor cell and replicate in the tumor cell. It would not be able to replicate well in a normal cell. And so we get that selectivity now from the virus doing its virus stuff. And we would actually kill that tumor cell in that way. There is a great 60 minutes piece on a clinical trial using polio to kill glioblastoma cells in patients that I have here. I cannot show you an entire hour-long 60 minutes clip right now. And there are a number of clinical trials of a number of different viruses doing this. But when people have actually started to examine what's going on with oncolytic virotherapy a little bit more, they have realized that they were missing a piece of the puzzle. And in fact, what people have realized is that when we have the virus infecting the tumor, we might have some tumor cells killed by the virus, but we're also providing a whole lot of PAMPs. And we're inducing a whole lot of inflammation. And we're inducing a really strong immune response in that location, as we should when there is a microbe. And suddenly, much of that immunosuppression that the tumor had induced gets broken. And suddenly, what used to be an area where we had immunosuppression now has inflammation as a result of that infecting virus. And now that immune response that used to be able to eliminate the tumor and sort of stopped because of immunosuppression is no longer suppressed. It's sort of woken back up. And we can start to have tumors killed around the body, sort of getting rid of those tumor cells, either because of directly lysing the tumor cells or because of actually inducing PAMPs, inducing other inflammatory responses, giving this a break in the tolerance that the tumor may have set up, while the normal cells are remaining unharmed. And I might be able to pull this off, maybe, possibly, maybe not. Is this gonna be it? Not what I wanted. Not it. You're not gonna let me paste? No paste. Select, select all. Come on. Anyway, this is a really great little video that I was going to end us off with, that I may or may not be able to end us off with. But it was talking about the virus modifications and the ways that they set up oncolytic virotherapy. And as someone who both geeks out about viruses and the immune system, you can imagine why I think it's really cool. I'm not sure I'm going to be able to pull this off. So I'm going to say we're done with that at this point today. So remember, you have the homework due on Friday and a review session on Saturday at 10. And I'm still happy to answer any questions from you or from this guy back here. I mean, the tumor likely is always going to win, but we're coming up with better ways to fight back. We can fight better than we could before. Maybe eventually the tumor won't always win. So anyway, that's what I got. So thanks, guys.